Again, my name is Enamul Huq, and I'm in the Molecular Biosciences Department at the University of Texas at Austin. This is our campus, UT Austin, the main administrative building. And if you come to Austin now in Texas, the Texas flower called bluebonnet. So all the highway sides, they sort of, there are lots of these wildflowers. So it looks beautiful. And this is the city where we live in. So as Jebapa mentioned, I'm going to talk about actually a slightly different topic. It's a new area in my lab. And my title is Global Warming and its Impact on Plant Development. Sort of my talk is at the perfect time, but right. So at the perfect time, I think most of you listened to Dr. Abed Chaudhary. So he mostly talked about climate change and global warming. So if you remember his talk, how sort of important topic is this? And this is highlighted on this slide. This is actually from Science this year. This is the temperature change over time. Scientists started measuring around this time, and you can see last 20 years, it's sort of in an upward trajectory. And from here, you can see it's about 0.5 degree difference now. But this is already causing alarm throughout the world, and especially people who understand. And obviously, those of you who follow US politics, you know there are lots of skeptical people who thinks this is a hoax. But people living in Bangladesh especially, they are affected on a day-to-day basis, right? So, sorry, it's, okay. So we see this kind of picture nowadays in the news, that there are lots of wildflowers throughout the world, and also polar bears are sort of not finding food at the right place. So because of this, understanding the effect of global warming is very important, very critical now. And it profoundly affects not only plant growth, it actually affects the whole ecological system. One of the impacts of global warming on plants is plants become taller. And when they are elongated, then they lodge, even with a small amount of wind, and there's a huge crop loss because of that, okay? And because plants are the primary producers, it's sort of at the bottom of the food chain, and all of the life forms on Earth depends on those. So that's why it has a cumulative impact. One of the impacts of global warming is plants flower early. And when they flower early, but the pollinators are not available at the right time, then there's no fertilization. So that way also crop loss happens, okay? So it's a profound impact. And a lot of labs are working on sort of understanding molecular mechanism, how this ambient temperature or high ambient temperature controls plant growth and development, and how we can make crop plants that are resilient to this increase in temperature. So I want to, I'm showing you all these big picture slides, but in laboratory conditions, what we use is completely artificial. We use sort of small growth chambers like this, where we use normal growth conditions around 20, 22 degrees. And when it is 35 to 45 degrees, this is called heat stress or high temperature. But for global warming research, people use more around this 26 to 30 degrees. This is probably common in Bangladesh right now. It's probably close to 30 degrees. But we use Arabidopsis as a model plant for research. So this temperature is already pretty high for Arabidopsis. Okay, so all of these are laboratory conditions, and translating this research into crop plants and ultimately making crops resilient to high ambient temperature, that's completely a different ballgame. And I want to mention that our lab is not focused on that. We are more focused on fundamental understanding of how temperature affects growth and development. And hopefully this basic understanding can be translated in future into crop plants and make sort of resilient crops that can be cultivated at higher temperatures. So what are the phenotypes that we see? So we work with Arabidopsis exclusively, and this is an Arabidopsis plant at the seedling stage. This is probably 1 week old. And you can see if we grow at 20 degrees Celsius, it has this architecture or morphology. If you grow at 28 degrees, notice there is a big change in this part of the tissue. This is called hypocotyl. Also, the leaves are more upward angle. The petiole is elongated. This is much older sort of before flowering stage. This is vegetative growth at 20 degrees versus 28. And you notice the petioles are longer, leaves are narrower, the upward angle for petioles. All of these are sort of for a reason. Plants sort of, this is an adaptive mechanism to cope with high ambient temperature. So these plants, they produce less stomates so that at high temperature there is less transpiration. It can conserve water. But obviously there is a trade-off. If you close the stomates, then you are not going to photosynthesize that much. Carbon dioxide is not going to come into the cells. This erectile position is because if you have this condition, the warm air gets trapped in the leaves. But if you have this upward angle, then the aeration can happen easily, and that helps cool down local temperature. Ultimately, this plant is going to flower early. That is also an adaptive mechanism because plants, when under a stress condition or unfavorable growth condition, the last thing they will do is produce some seedlings, produce some seeds that will continue for next generation. Okay. So early flowering is another phenotype for high ambient temperature. So all of these collectively called thermomorphogenesis, sort of at plant responses at higher temperature. Okay, so what are the sensors for high ambient temperature? So far there are 3 temperature sensors identified. So one, the first sensor identified is called phyB, phytochrome B. So this is the photoreceptor that we work with in our lab. These are light receptors. In the dark, they exist in an inactive form called PR form. If you shine red light, they photoconvert, sort of change the conformation of the protein into a PFR form. And the PFR dimer is the biologically active form. It moves into nucleus. It binds to these phytochrome interacting factors and also many other proteins and controls their function in different ways. Now, this PR to PFR form, it exists in an equilibrium. Okay, it's a thermodynamic equilibrium. And that equilibrium is controlled by temperature. So it's almost impossible to make 100% PFR. If you constantly expose to red light, it will make sort of 95% or 98% PFR. But some molecules will instantly go back to PR because that's the thermal relaxation process. Now that process is enhanced by warm temperature. So this is the molecular basis for temperature perception by phytochromes. That at high temperature, phytochromes are in active form. At low temperature, phytochromes can be active form and display its molecular function. And so this is the first receptor identified. The second one identified is sort of a more prokaryotic type. This has been shown in bacteria where sort of hairpin loops in mRNAs that functions as temperature sensors. So this, with a large hairpin loop, ribosome cannot bind properly and cannot translate phyB7 mRNA. But at high ambient temperature, this stem loop structure is resolved. Ribosome can bind, find the correct ATG, and initiate translation, and the phyB7 can move into nucleus and control thermomorphogenesis. So this is the second type. And the third type recently shown another protein called ELF3, is called early flowering 3. So at lower temperature, this protein is a monomer that binds to phyB4 promoter and controls phyB4 expression. But at high ambient temperature, ELF3 forms this phase-separated complex. I think many of you probably know this now, that many proteins sort of undergo phase separation and forms this aggregated complex. And these are the aggregated complex at high ambient temperature, and it cannot bind to phyB4 promoter, cannot control. And so these are, so far there are 3 ways plants can perceive higher temperature. Okay, there's a nice review came out this year in Plant Cell Environment that summarized all these studies. Now, what are the factors downstream of these receptors or thermosensors? So one of the factors identified is called phyB4. This is discovered in 2009, Coini et al. They showed that wild-type plants that respond to high ambient temperature, notice here, elongated hypocotyl, longer petiole, upward angle, but phyB4 mutant does not show any phenotype. Right here, phyB4 mutant. Now, phyB4 is part of a family member. In fact, most of them were originally discovered in the previous lab that I worked as a postdoc at UC Berkeley. So these are PIFs, there are 8 PIFs, and all of these are basic helix loop helix transcription factors. So these factors have a basic domain, and then they have a helix loop helix. And this helix loop helix is a dimerization domain. So these two proteins dimerize like this, and then they bind to DNA. And that's how they function. So PIF4 as a transcription factor that binds. PIF4 protein stability is controlled by high ambient temperature. It is stabilized. So downstream of the thermosensor, PIF4 is the main hub transcription factor that controls these phenotypes, thermomorphogenesis. Now, because we are mainly focused on light responses, and we have previously shown that like phyB forms a complex with PIFs and also another protein called SPA, suppressor of phytochrome A. And this complex results in degradation of PIFs. So this is shown for PIF4. Notice in response to light is getting degraded. But that is under red light condition. But at high ambient temperature, PIF4 is stabilized. Okay, so this is the opposite effect. Now, so this is the sort of the model when we started working on these, that phyB is the main thermosensor that inhibits PIF4, and when the phyB is inactivated at high ambient temperature, PIF4 is active, binds to E or G boxes, and it activates genes that are involved in auxin biosynthesis as well as auxin signaling. And the increased auxin level and signaling, that actually promotes all this elongation growth. Okay, so that has been shown a while ago. Now, our interest is that we showed in 2019 that this complex forms, these 3 protein complexes, and that results in degradation of PIFs. So in thermomorphogenesis, scientists have shown role of phyB, PIF4, but nobody knew what's the role of SPAs. And we know they function in light signaling pathway. So we decided to look at whether SPAs also play roles in thermomorphogenesis process. But while we are working on SPAs, we also decided to look at sort of more comprehensively thermomorphogenesis phenotypes, because in the past, people mostly focused on the aerial part, sort of above ground tissues for thermomorphogenesis. People sort of mostly overlooked the below ground or root elongation phenotypes. So we started looking at sort of more globally, what's the response of plants. And I'm not sure if you can see it. Notice these plants, they are longer hypocotyl, but their hypocotyl is also thicker. So there is a general sort of growth stimulation. Notice the roots are much longer than 22 degrees. Hypocotyl is thicker, hypocotyl is longer, petiole longer. So all of these phenotypes that we were looking at. So I'm going to tell you 3 stories. 2 are mostly focused on the shoot part. And the 3rd story is more focused on the root part. And I'll show you sort of a new, not a new, sort of previously known factor playing a role in root thermomorphogenesis. Okay, so let's start with the SPA proteins that I showed in the previous slide. So there are 4 SPA genes in Arabidopsis, and these are relatively large protein, more than 100 kilodalton. They have a kinase domain at the N-terminus, and I'll show you the pictures or structures. And it has WD40 repeat, also ring finger domain, ring domain, or coal domain. So we have a gauntlet of mutants, these SPA mutants, sort of single, double, triple, quadruple mutants. Okay, and also complementation lines. So notice this is the wild type, shows the elongated phenotype, SPA1, almost like wild type, very little difference. As you go into double mutant, we see now hypocotyl is shorter than the wild type. When you go to quadruple mutant, notice these plants are not even responding to high ambient temperature. Okay. Now, if we put back just one SPA out of 4, SPA1, into the SPA quadruple mutant, it can partly rescue the phenotype. Okay. And this is the overexpression phenotype. Overexpression phenotypes for overexpression lines. It's losing the okay. Okay, so then we did RNA-seq studies to identify sort of gene expression difference between wild type and spaQ at 2 different temperatures, 22 versus 28. And you can see in wild type, there are lots of genes that are uniquely regulated. Those are SPA dependent. So these genes can be classified into upregulated or downregulated. And you can see almost more than 2,000 genes are upregulated in wild type, or similar number is also downregulated in wild type. Now, many of the marker genes in thermomorphogenesis are shown here. As I told you before, targets many auxin biosynthetic genes or signaling genes. IA29 is a signaling gene. And notice temperature is a stimulating expression in the spaQ background that is absent. SAUR15 is another auxin signaling gene that is also upregulated, and in spaQ it is downregulated. PRE is another transcription factor. CYP79B2, that's also involved in auxin biosynthesis. So all of these genes are showing stimulation in response to a temperature, but that stimulation is absent in the spaQ background. So SPA proteins regulate large numbers of genes at high ambient temperature. So we wanted to look at the genetic relationship between PIF4 and spaQ. Again, this is the spaQ not showing phenotype. Notice PIF4 shows exaggerated phenotype because PIF4 promotes thermomorphogenesis. But if you put PIF4 in the spaQ background, spaQ is epistatic. This strong elongation phenotype is almost gone here, eliminated. phyB mutant shows this elongated phenotype. If you put them in a spaQ background, it is suppressed. So spaQ is functioning epistatically with both PIF4 and phyB. It suppresses their phenotype. Now, when we look at PIF4 and phyB protein level in a spaQ background, notice here at high ambient temperature, PIF4 is stabilized, but in the spaQ background, the protein is absent. So protein is unstable. But on the other hand, phyB is more stable at both sort of high ambient temperature and normal temperature. So the fact that PIF4 phenotype is gone in a spaQ background, that could be because of absence of the protein, okay? So SPAs are necessary for stabilization of PIF4 and destabilization of phyB, sort of at the protein level, post-translationally. Okay, so this is the story we published in 2019. So this is the structure of SPA protein. It has an N-terminal kinase-like domain, coil-coil, and WD40. Within the kinase-like domain, and there are some amino acids that are highly conserved. And we actually created a single amino acid that arginine is highly conserved among all these plants from various SPA proteins. And we created single amino acid arginine to alanine. And that actually abolishes about 70% of the kinase activity. Okay. So, all these projects actually is done by a single graduate student. His name is Sanghua Lee. He defended actually last Friday. Right, it's last Friday. So he's graduating this semester. So he did most of these work. So he showed that this is the SPA1 autophosphorylation and transphosphorylation to P4. And then if we use the mutant SPA1, we see the transphosphorylation is reduced or eliminated. So SPA1 can directly phosphorylate P4, and that phosphorylation is important for SPA function and also regulation of P4. So this is shown here. Again, this is the wild type. SPAQ does not show phenotype. Now, this is the SPAQ. If we put SPA1 into SPAQ background, notice it is rescuing the phenotype almost 70%. But if we now introduce the mutant single amino acid change SPA1 into SPAQ background, it does not rescue the phenotype. Okay, so at both temperature, there is no change. So the SPA1 kinase activity is essential for thermomorphogenic phenotype. Now, if we look at P4 protein level in these plants, notice P4 is stabilized at high ambient temperature. And you can probably see double band here. So we think these are, one is the top band is phosphorylated, bottom band is unphosphorylated. And in the SPAQ, it is absent. If we introduce SPA1, that largely restores P4 level. But if we introduce the mutant SPA1, P4 level is still very low, and the phosphorylated band is missing. This band is unphosphorylated form. Now these bands, we can stabilize it using a proteasome inhibitor. We use routinely called bortezomib, or many of you might have used MG132 or MG115. So those are called 26S proteasome inhibitors. And if we inhibit, notice here, in the mutant SPA, at 28 degree plus bortezomib, we only see the unphosphorylated form. So, we think that SPA proteins are directly phosphorylating P4, and that phosphorylation is necessary for stabilization of P4, and that promotes thermomorphogenesis. Okay, so it's just to summarize this part, so far what I showed, sort of new function of SPA proteins, that SPAs are essential for thermomorphogenesis, and the way SPAs function, they stabilize P4 and destabilize phyB. And then finally, the way they stabilize P4 is by directly phosphorylating P4 and preventing proteasome-mediated degradation. That way, phosphorylated phyB4 is more stable, that can bind to the sort of growth gene promoters. And mostly these are auxin signaling and biosynthetic, and that promotes sort of elongated growth. So this story was published last year in Development. Any question on this? Or we can continue and ask questions at the end.
So I think
you can stop me at any time and ask questions.
Okay, I think we can take the questions later.
Okay, awesome. Okay, so now the second story is actually came out of the first story. Remember, we did these RNA-seq studies using spaQ and wild type, and we're looking at these about 2,000 genes that respond to temperature in wild type, but not in spaQ. And two of these genes that looked familiar because we worked with these genes in the past, these are called HEC proteins, okay? So these HEC proteins are upregulated in response to high ambient temperature. Notice here, this is again a confirmation of RNA-seq using qRT-PCR. HEC1 is upregulated and HEC2 is also upregulated. We had HEC2 GFP transgenic plant expressed from 35S promoter. So we looked at the protein level and noticed the protein is also upregulated at high ambient temperature. Now the HEC proteins are again familiar because they are part of this large basic helix loop helix family, except that these proteins, they do not have this part, the basic part. They have helix loop helix without the basic domain. So what they do is they can homodimerize and heterodimerize. And if they heterodimerize with a basic helix loop helix, then it actually makes the BHLase protein inactive because it cannot stably bind the DNA. For these proteins to bind DNA, they have to bind this palindromic sequence on both sides of the DNA. So when the heterodimer sort of BHLase and HLase, then one part of this is missing, so it cannot bind to DNA. Now, HEC proteins have been shown to function in flowering transition. We showed in light signaling, shoot meristem, Chinesium development. So a number of labs working on these proteins. So in 2016, we showed that there's a feedback negative loop present between PIFs and HEC proteins. So PIFs actually directly bind to HEC promoters and activate HEC mRNA that translates into protein that actually binds to PIFs and form this BHLase, HLase heterodimer, and that cannot bind to DNA. So HECs, they are controlling their own expression by controlling the activity of the PIF proteins, okay? Where PIFs are transcriptionally activating and HECs are post-translationally controlling PIFs activity. So that sort of feedback loop we showed previously. Now, we had all these materials like mutant overexpression lines. So we looked at the HEC2 overexpression plants, and it shows sort of attenuated thermomorphogenesis phenotype. This hypocotyle is shorter than this wild type. Notice in the double mutant background is now much taller than the wild type. So again, HECs are negatively regulating thermomorphogenesis, whereas PIF4 is promoting thermomorphogenesis. So as I showed before, the previous research from other labs, they focused on PIF4. And now we looked at all other PIFs because our lab has all the PIF mutants and overexpression lines. So I'm showing the data for overexpression. The mutant also shows similar sort of opposite phenotypes. So notice PIF1, 3, 4, 5. These are the 4 major PIFs. And they're all overexpression lines show exaggerated thermomorphogenesis phenotype. If we look at the protein level for all 4 proteins, this is 22 versus 28, 22 versus 28. All 4 are stabilized in response to high ambient temperature. So we think it's not only PIF4, it's all the PIFs are contributing to thermomorphogenesis phenotypes. So we looked at more genetic relationship between HECs and PIFs. Notice the double mutant shows exaggerated phenotype. Now, if we create hexaple mutant, HEC1, HEC2, and PIFQ, it looks more like PIFQ. So PIFQ is epistatic to HEC1, HEC2. Okay. Now, again, this is HEC1, HEC2, and this is PIF4 overexpression. Notice in the PIF4 overexpression in the HEC1, HEC2 background, now it shows even a stronger phenotype. This is because in the absence of HEC1, HEC2, PIF4 activity gets increased. There's no helix loop helix to inhibit PIF4 activity. That's why PIF4 shows a stronger phenotype in the HEC1, HEC2 background. Again, we did RNA-seq studies and sort of compared 3-way comparison, wild type, HEC1, HEC2, and PIFQ. And we dissected sort of PIFQ dependent or HEC1, HEC2 dependent genes. And we found, again, the marker genes for thermomorphogenesis. This is in auxin biosynthetic pathway. Notice it goes up in response to high ambient temperature, but in the HEC1, HEC2 background, it goes up much higher because this is a PIF target gene and the PIF activity is higher in the HEC1, HEC2. In the PIFQ background, this regulation is gone. This is auxin signaling gene. Again, in wild type, it goes up. HEC1, HEC2 background, much stronger upregulation. PIFQ background, no regulation because this is a PIF direct target gene. So just like HEC1, HEC2, there are other helix loop helix proteins. HFR1 is another helix loop helix and also a PIF target gene. Notice this one also upregulated, and in the HEC1, HEC2, it much stronger. In the PIFQ background, this is much reduced. PIL1 is another helix loop helix, same regulation. These are all PIF target genes. Okay, so now again we look at protein levels of PIF4 or phyB, and notice in the Columbia background, PIF4 is stabilized, but this is native PIF4 we are looking at, native PIF4 antibody. And in the HEC2 background, PIF4 is even more stabilized. In the HEC1, HEC2, less stabilized. We showed that in the HEC1, HEC2 background, there's a faster degradation of PIF4 compared to just PIF4 background. If this is a transgenic plant, PIF4 MIC. So HEC1, HEC2, they are heterodimerizing with PIF4 and preventing PIF4 degradation. It's stabilizing, but you will see that it is stabilizing but preventing its function because it's a heterodimer. So even though there's more PIF4 protein here, But these plants don't show the phenotype because the PIF4 activity is reduced because of heterodimerization. Okay, so I want to show you more directly if that is the case. So we have shown in the past, this is the basic domain for BHLase proteins. This is the helix 1 loop, helix 2. Within the helix 2, there are two amino acids that are highly conserved. This methionine and leucine. If we mutate these two residues to D and E, aspartic acid and glutamic acid, then this sort of cannot heterodimerize or homodimerize these proteins. Okay, these just two amino acid mutations. Okay, so notice now we're looking at HEC2 interaction with PIF4 in these two hybrid assay. Wild type HEC2 interacts with PIF4. And if we introduce the two amino acid mutant, it cannot interact. We have transgenic plants expressing wild type HEC2 and mutant HEC2. Wild type HEC2 can immunoprecipitate PIF4 MIC. Notice here, this is the PIF4. We are pulling down with PIF4 and blotting with GFP, showing HEC2 GFP. But PIF4 cannot pull down the mutant HEC2. So this is sort of showing in vivo co-IP assay. Now, what's the phenotype of this? Is HEC2 overexpression plant that shows attenuated phenotype, but the mutant HEC2 shows more like wild type phenotype because the mutant HEC2 is not functional anymore. Okay. So PIF4 HEC heterodimer formation is essential for repressing thermomorphogenesis. Okay, and among all the PIFs, PIF4 is the only PIF that actually binds to its own promoter and controls its own expression. So PIF4 directly binds to the PIF4 promoter and activates PIF4 mRNA. Okay, so this is shown here. In response to high ambient temperature, PIF4 is stabilized and that activates its own expression. In the HEC1, HEC2 background, you see much more expression because now PIF4 activity is higher. We can show this by cheap assay called chromatin immunoprecipitation assay. At high temperature, PIF4 binding to its own promoter is stimulated, but in the HEC1, HEC2 background, it's even higher because HEC1, HEC2 is not inhibiting PIF4 binding anymore. So this is we call sort of autoregulation of PIF4 by itself. Now, as I showed you before, that PIFs, all PIFs, they actually bind to HEC promoter. So all these PIFs binding to HEC promoter, activating HEC transcription, and then in turn, HEC interacts with PIFs and inactivates them. So this is the negative feedback loop. So notice in the wild type, HEC1 is stimulated, but in the PIF4 background, that stimulation is attenuated. PIFQ background is gone. HEC2 shows the same behavior. If we look at now PIF4 enrichment, like if this is PIF4 binding to the HEC promoter, notice PIF4 binding is stimulated at high ambient temperature. So at high ambient temperature, PIF4 is more stable, that binds to HEC promoters, activates HEC genes, and then HEC in turn inactivates PIFs. So overall, this PIF HEC, they are forming an autoregulatory negative feedback loop that controls thermomorphogenesis. So HECs are transcriptionally and post-transcriptionally increased at high ambient temperature, and they inhibit thermomorphogenesis through PIFs. We also show that all major PIFs contribute to thermomorphogenesis, and this heterodimerization actually forms this autoregulatory negative feedback loop, where PIF4 transcriptionally activates HECs. HECs actually stabilizes PIF4 post-translationally, but then negatively regulate PIF4 activity. And then PIF4 controls its own expression. So whatever resulting PIF4 homodimer is present, or other PIF homodimer, that is actually activating the growth genes that controls thermomorphogenesis. Okay, so this is sort of the summary of the second part of my talk. And I'll go to the third part, as hopefully you will remember this so you can ask questions at the end. So as I showed you before that, we are looking more sort of comprehensively this thermomorphogenesis phenotype, both above ground and below ground. Majority of the studies, except one or two, in fact one, majority of these in the field, they focus on above ground, like shoot phenotype, not root phenotype. Only one lab has shown that root is elongated, and they showed that it's the prasinosteroid pathway that controls root elongation. Now we looked at root elongation over time, sort of under short day, long day, continuous light in different conditions. And we find that the root is elongated. Now, PIF4 is the main hub for thermomorphogenesis. So we look at root phenotype for PIF4 mutant and PIFQ mutant. Notice both PIFQ and PIF4 show elongated root, okay? So we hypothesized that there must be another regulator that controls root phenotype. PIF4 is not the only regulator. PIF4 is mainly focused on shoot part. So again, we have lots of like signaling mutants. So we looked at many of these, and we found that one, so this is wild type showing elongated root. HY5 is a BGIP transcription factor, and its role in light pathway is well established for the last 20, 30 years. And notice here, it does not show that root elongation phenotype. Again, PIFQ shows phenotype. If you make HY5 PIFQ, it does not show phenotype. phyB is the master regulator, so phyB controls everything. It does not show the phenotype. phyB GFP shows elongated root. So we think that HY5 is the main sort of transcription factor that is controlling the below ground phenotype. Okay. So again, we did RNA-seq studies using samples collected from the shoot part and root part. And we compared between wild type and HY5 at 2 different temperatures. And you can see many HY5 dependent genes in both root and shoot. And these are shown in the heat map. Lots of genes show altered gene expression. And now, recently, this is actually in press in New Phytologist. We showed in our lab that SPA proteins can directly phosphorylate HY5. So this is the kinase assay that shows this is the same amount of SPA protein with increased amount of HY5, shows much higher level of kinase activity. And we mapped the kinase phosphorylation site to a single amino acid, the serine 36. We mutated that to create 36A and 36D. D form is the constitutively sort of mimicking phosphorylation site, and the A form is phosphonull site. So we have all these transgenic plants, and we recently published this, And we showed that if you mutate that single site to alanine, you can eliminate a majority of the phosphorylation. Basically, it's gone. It's the background level. So we had all these transgenic plants. So we look at the root elongation phenotype. Notice wild type shows root elongation. This is the HY5 mutant that does not show root elongation. Now, wild type HY5 complemented into the HY5 mutant background that shows root elongation phenotype. And this is the D form that also shows longer root, but the A form cannot show the longer root. Okay. In fact, HY5 A form is a more active form of HY5. It binds to DNA more efficiently, but this A form cannot rescue the root elongation phenotype. Okay, so phosphorylation HY5 is essential for root thermomorphogenesis. And one way for this is, again, we look at stability of protein. So notice at 22 versus 28, this is HY5 GFP. It's much more stable at 28. We have Western blot data. I'm just showing you the GFP blots. Both wild type and D form is stabilized, but the A form is stability is reduced at high ambient temperature. So HY5 protein is also post-translationally regulated by high ambient temperature. So now we go back to the PIF lines, the SPA lines that, so this is the wild type showing longer root. spaQ does not show longer root. If we introduce wild type SPA1 into the spaQ background, that shows now elongated root. If you have single amino acid point mutant SPA1 introduced into spaQ, that does not show longer root. So again, the kinase activity of SPA proteins are necessary for root thermomorphogenesis. Okay. So we also looked at regulation of these proteins, sort of why PIF4 controls above ground response and HY5 controls below ground response. Okay, so we looked at expression of HY5 and PIF4 in a tissue specific manner. So we looked at, this is QRT-PCR in the whole seedling. Notice only in the root, HY5 expression is stimulated. Okay, but if we look at PIF4 in the whole seedling, it is stimulated. In the shoot part, it is stimulated, not in the root part. So PIF4 expression is stimulated in the shoot part. This is at the gene expression level, and HY5 is stimulated at the root tissue. If we look at native PIF4 and HY5 protein level, notice this is PIF4 at 28 degree, is stabilized in the root shoot sample, not in the root sample. But if we look at HY5, HY5 is stable in the root sample, not in the shoot sample. Okay, so these genes are spatially regulated, both transcriptionally and post-translationally. So this is probably one of the reasons why they are dedicated to different tissues, like PIF4 for above ground tissue and HY5 for below ground tissue. So in summary, again, my third part I want to summarize here, that high ambient temperature promotes this root elongation and shoot elongation, and PIF4 plays a critical role. PIF4 is essential for this promotion. And now phyB negatively regulates PIF4. phyB is the master regulator because that's the sensor for high ambient temperature. So it negatively regulates PIF4, but it indirectly stabilizes HY5 both in the shoot and root part, okay? And SPA proteins are functioning in both root and shoot. And one of the ways they function is by directly phosphorylating HY5 and stabilizing in the root tissue, and also PIF4 in the shoot tissue. And PIF4 by itself is controlling hypocotyl elongation. I did not show you data that PIF4 and HY5 both, along with the SPAs, they control actually hypocotyl thickening. And mainly HY5 controls root elongation, the below ground phenotype. So both shoot and root respond to high ambient temperature, and there is a spatial regulation of thermomorphogenesis by sort of these selective transcription factors. Plants are employing different master regulators for controlling different tissues, specific responses. And the kinase activity of SPA proteins are essential in both tissues, both above ground and below ground. Again, this story is under revision in Nature Communication. Hopefully this will be published soon. So overall, just to summarize what I showed you, that in the introduction I talked about, the high ambient temperature has a profound impact on plant architecture. And this is shown here in the model plant Arabidopsis, but many other labs are working on crop plants and they see a similar impact. And we show here that the kinase activity of SPA proteins are essential for thermomorphogenesis, where SPA1 directly phosphorylates both PIF4 and HY5 and stabilizes them. PIF4 and HY5 are main sort of master regulators in the shoot and root tissues, respectively. We also show that for other PIFs, major PIFs, they also contribute to thermomorphogenesis. And finally, we also show that the HEC and PIF4 forms an autoregulatory feedback loop that controls thermomorphogenesis. So this is sort of the overall summary of my talk, and I want to acknowledge, as I said before, all three of these projects are actually initiated by a single graduate student, Sang-Hwa Lee, and he just graduated last Friday. He already got 2 postdoc offers. He declined one, and he's going to Salk Institute, probably end of May. So this is the current lab, and these are all previous postdocs and graduate students. They contributed to some of these studies. We collaborate with large numbers of people on different aspects. And also we acknowledge funding from different sources like NIH, NSF, and BSF. And I will be happy to answer any questions.